Hello, everyone. Welcome back to Hang Education, where we share the knowledge of biology. So today in this video, I'm going to share my sample answers to the current February-March 2025 biology theory paper, which is paper 4. So let's begin. So starting off with question number 1. So it's a figure 1.1 show the movement of particle through an epithelial cells in the small intestine. So they give substance P, Q, and R. And as we can see from the diagram, so for the substance or for the particle P, it is moving from high to low concentration. And for the substance R, also it is moving from high to low concentration. But for the particle or substance Q, they are moving from low to high concentration using energy. So this is just, this already give you a hint of like the type of transport system involved in this mechanism. So anyway, for the number A, so it's a figure 1.1. Arrow P represent the diffusion, and it's asked to define the term diffusion. So this is the definition. So diffusion is the movement of particles or molecules from a region of higher concentration to a region of lower concentration, which is down a concentration gradient due to the random movement of a particle. So this is a very typical definition that every student should be familiar with. And next one is the type of energy needed for diffusion. So for the diffusion, they are due to the random movement, so which is by the kinetic energy of the molecule itself. So the type of energy should be kinetic energy. And number B, carbon dioxide molecules also move by diffusion, and state the name of the process in human that produces carbon dioxide. So the process that produces carbon dioxide in the human cell is respiration or aerobic respiration or cellular respiration, right? So that's why the answer is cellular respiration or aerobic respiration. It's also defined. And figure 1.1, draw an arrow. So basically, we have to draw an arrow to show the direction of diffusion of carbon dioxide molecule. So here, you need to be very careful because the question is asking diffusion of carbon dioxide molecule. So carbon dioxide is the product of respiration, and it have to move out of the cell. So from my assumption, so I drew an arrow that is moving out of the cell. So what I assume is that respiration will take place inside the cells and carbon dioxide will be produced. So the carbon dioxide as a product, it will released out of the cell. So I drew an arrow that is going out of the cell. So this is my answer to this question. And then in figure 1.1, arrow Q represent another type. And as we already seen that the arrow Q is showing from low to high concentration. So the type of movement involved would be the active transport. And the explanation is the particles are moving from lower to higher concentration against the concentration gradient, and it uses energy from the respiration. So this is my answer. And also in figure 1.1, particle R move from the lumen of the small intestine into the epithelial cells and suggest why particle R cannot be starch. So you have to remember, starch is a polysaccharide, which is a large molecules, and they cannot be transported by diffusion. So starch molecules are very large and cannot diffuse or pass through the membranes, right? Since they are a type of polysaccharide, and it must be broken down into smaller and soluble molecules such as glucose by amylase before it can be absorbed. So basically, after we eat starch, it have to be broken down into glucose by the enzyme amylase, then only it can be transported, not the starch, but the product of the digestion, which is the glucose, right? So this would be my assumption answer. And number E, figure 1.2 is a photomicrograph of red onion cells. And as we can see from the diagram itself already, it is a state of turgid, and it have the turgor pressure, or the cells are in turgid stage. And in figure 1.3, it said that the same red onion cells, they are immersed in a salt solution. And we can see that the cells are in a stage of flaccid, or it can also be plasmolized, right? Because we can see that the cell membrane and the cell wall, they are not attaching, and we can see a lot of cytoplasm spaces and telling us that the water have moved out of the cells by osmosis. So this is just the assumption before we go into the answer. And okay, so now we can see that figure 1.2 and 1.3 describe and explain. So you have to be careful. It's a describe and explain. So you cannot just describe the appearance of the cells from the diagram. You also need to explain the difference in the appearance. So as we already discussed a bit, right? So in figure 1.2, the cell walls of the red onion cells are intact, so which means they have turgor pressure, which presses against the cell wall and maintaining their normal shape. So this indicates that water entered the cells from higher water potential to lower water potential through partially permeable membrane by osmosis. However, after the cell has been immersed into the salt solution in figure 1.3, the cells become shrinkens, in which the cell membrane has pulled away from the cell's wall, losing their turgidity. And when they are immersed in the solution, which has the lower water potential than inside the cell, so the water molecules move out of the cells from higher to lower water potential by osmosis, which result in the shrinkage or the flaccid. And gradually, the cells can lose their turgor pressure and it will become plasmolyte. So basically, you have to describe the appearance of the cells and then you have to explain why this appearance of the cells are provided. So to be summarized again, so figure 1.2 is show the stage of turgidity, showing that the water has entered the cells from higher water potential to lower water potential, and it has a turgor pressure which presses the cell, which press the against cell wall and maintaining their normal shape of the cells. And when they are immersed into a salt solution, so salt solution will have lower water potential. So the water moved out of the cells from higher to lower water potential by osmosis, and which lead to the state of flaccid, and it can lead to the plasmolyte situation, right? So this would be my sample answer to these questions. So question number one is done. And question number two is that the internal body temperature of a person was recorded, and figure 1.2 showed the result. And as we can see, the graph is from 36 to 38 temperature, and then we can see the time. And number one, it's asked calculate the temperature range. So it is asking the temperature range, right, for the internal body temperature, and you have to state the unit. So the highest point is 38 and the lowest is 36. So I subtracted that too. So 38 subtract 36, so which give 2, and 2 unit is 2 degrees Celsius. And on figure 2.1, draw a line to show the set point, right? So the set point is normal body temperature of the mammals or of a person is 37 degrees Celsius. So I drew a line through a 37 degrees Celsius. So that will be a set point, right? And then state the name of the mechanisms for homeostasis control. So it's simple. So it is negative feedback. And then region F show a change in body temperature. So be careful, it's that region F show a change in body temperature. And when we look at it back the graph, region F is showing the increase in body temperature. So basically, when the body detect that body temperature is below 37 and it start to increase, right? So we need to remember, we cannot just answer anything out of our mind. So it's showing the stage of increasing temperature. So we have to talk about the mechanism that to increase our body temperature. And it's that explain how the body causes the change in the body temperature shown. So my answer would be the thermoreceptor detect the drop in the body temperature, which is below 37 degree. So it will send the impulse to the brain and hypothalamus using sensory neuron, and the brain will respond to the effector, which is in this case the skin, through the motor neuron. And as a result, the arterioles will become constrict, right? So it's vasoconstriction, so reducing the blood flow to the near of the surface and reduce the heat loss by radiation, as well as at the same time, the hair erector muscles also contract, pulling the hairs to stand up, which will trap the layer of air as insulation. And also the muscle contraction and relaxation will become rapid. So it is shivering process. And at the same time, the rate of respiration or metabolism will be also increased to release heat energy. So these are all the mechanisms which our body perform to increase the body temperature when the body temperature is below the set point, which is the 37 degrees Celsius. So this is my answer. And then figure B, number B, figure 2.2 is show a cross section of a human skin. And number one, the letter, so you have to state the letter of the structure shown that produces sweat. So we have to look at it. So the way how I look at for this answer is like when I see the pore, so I like check the direction, and the label A is it possibly could be the sweat gland, right? So it will produce sweat, and then the sweat will go up through the pores and it get evaporated. So I chose letter A. And structure C and D. So structure C is the hair erector muscles, and structure D is nerve endings, or it can also be a receptor. So number three, moving on to question number three, figure 3.1 show the percentage of water in different structure of the human body. So we can see different body parts, the organ, and then they give you a percentage of water. And the mean mass of human eye is 28 gram, and you have to calculate the mass of water in human eyes. And be careful, they ask to give in two significant figures. So most students, if they do not read the questions carefully, they forget about the significant figures and everything, and they will just, straight away the answer. So you can lose point for that. So it said that for the human eye is 95%, right? 95%. So 95%, which is 95 by 100 into 28 grams. So we get 26.6 gram, and we need to convert it into two significant figures, so which is 27 gram. And number two, describe the importance of water. So these are my answers. So we learned that importance of water, or the properties of water, they are as a reactant, solvent, coolant, right? Transport, everything. But overall, in the human body, when I think about water, the first thing came to my mind is as a solvent in the blood plasma, transporting and carrying the nutrients and other substances, other soluble substances around the body. And it's also act as a coolant, right? As a sweating mechanisms where the water is involved. And also it is the component of the saliva and mucus, which helps in the digestion as well as keeping our respiratory pathway moist. And beside that, I also note down some point, right? Water is important in the cytoplasm of the cells as well. And it's also to maintain the balance of body fluids, right? So these are my answer, but these are all like most about the importance of water. But to be able to gain 3 points, you don't need to write everything. But these are my sample answer to this question. And then number B, cholera is a disease caused by pathogens. So what came to my mind, cholera, bacteria, right? And diarrhea and toxin and everything. And they show you a diagram of a cholera pathogens and name of structure L and M. So structure L is cell membrane and structure M is ribosome. And two features that are also found in typical prokaryote. So we can see that plasmid and flagellum, and also I thought about loop of DNA as well. And pathogen, type of pathogen that causes cholera is bacteria. And they give you a scientific name of the pathogens, and we have to state the genus. So it's Vibrio cholerae, right? So Vibrio is a genus and cholerae is a species. So Vibrio, and then don't forget to underline because since it is a scientific name. And explain how the cholera pathogens causes dehydration of the human body. So we have to talk about the process and explain, right? So cholera pathogen, it will produce toxin and it will attach to the intestinal lining, and it causes to secrete chloride ion into the lumen of the intestine. So when the chloride ion enter the lumen, so it will lower or reduce the water potential. So it will reduce the water potential. Then the water moved into the lumen by osmosis to able to balance the concentration. So as a result, there will be an excessive water loss in the form of diarrhea as well as dehydration, right? Because a lot of water will be lost in the form of watery feces, and also water will enter the lumen to be able to balance the concentration. So that will lead to the dehydration. So this is for question number 5. And question number 6 is about genetic modification. So during genetic modification, human DNA and plasmid DNA are cut with a restriction enzyme, which creates sticky ends, which are joined together by using an enzyme called ligase or DNA ligase. The modifying plasmid containing human genes is called a recombinant plasmid, right? So this is pretty straightforward and simple, I would say. And moving on to question number 4, figure 4.1 shows part of human placenta and umbilical cord. The arrow shows the direction of the blood flow. So we can see the placenta and umbilical cord. And be careful, it's that describe the function. So it's basically explain, right? Explain. Describe the function of the placenta and the umbilical cord. So we can separate it into 2 parts. So it's get 4 points. So you have to mention 2 points for placenta and 2 points for umbilical cord. So for placenta, it's allow exchange of substances between mother's blood and the fetus blood, such as like glucose, amino acid, oxygen, which will pass from mother to the fetus, and waste material such as carbon dioxide and urea, it will be passed from the fetus to the mother without the blood mixing together. And the umbilical cord contain blood vessels such as umbilical artery, umbilical vein, which connect the fetus to the placenta, ensuring the continuous flow of oxygenated blood carrying oxygen and glucose from the placenta to fetus, and also removing the waste substances such as carbon dioxide and urea. So make sure you need to include both about placenta and the umbilical cord. And then again, describe the function of amniotic sac and the amniotic fluid. So amniotic sac, which encloses and protect the developing fetus, and it also produces amniotic fluid. So amniotic fluid, which acts as a cushion, absorbing shock and protecting the fetus from the mechanical damages or injury. And it also helps to maintain the constant environment. So which basically providing the sterile environment that is optimum or suitable for the fetus to be able to develop inside the mother uterus. And then syphilis is a sexually transmitted infection, STI, that can be passed from mother to her fetus. And you have to state name of one other STI. So HIV or human immunodeficiency virus. And 2 ways to control the spreads of STI is by using condoms and also by limiting the number of sexual partner. And also we could mention about by not sharing the contaminated needles, right, while using the drug. So like sharing needles also can be another way that STD or STI can be spread from a person to another. And moving on to question number 5 is figure 5.1 show a cross section of a leaf, and then on figure 1.1 draw a circle around one vascular bundle. So we can see I drew a circle around a vascular bundle, and then a label line and letter H to identify palisade mesophyll tissue. Oh, I forgot I drew a line, but I forgot to write a letter H. So you have to identify a palisade mesophyll cell, and you have to write a letter X. And state the name of cell structure. So I would like to remind to read the questions carefully. Sometimes students lost point for not reading the instruction carefully, and they just answer whatever they want to. So that's how it leads to like reduction in the point. So name of the cell structure in palisade mesophyll cells where photosynthesis occurs. So this is very simple and pretty straightforward. So it's chloroplast. And describe the function of the tissues in a vascular bundle. So it's that tissues in vascular bundle. So vein or vascular bundle contain 2 different types of tissues, xylem and phloem. So you have to mention about xylem, the function of xylem and phloem, right? So xylem is transport water and mineral ions from the roots to other part of the plants, and it's also help to support. So basically, both tissues, right, xylem and phloem, they help to support the plant as well as the structure of a leaf. And for the phloem, it transports sucrose and amino acid from source to sink. So overall, both xylem and phloem in the vascular bundle, they also act as a support system, and also xylem to carry water and mineral from the roots to all part of the plants, or especially to a leaf, and phloem is transport sucrose and amino acid from source to the sink. And then question number B. So leaf shown in figure 5.1 is formed from an aquatic plant. And for number B1, state the term used to describe plants that are adapted to live in water. So when we study about adaptive features in biology, we mention about two types of plants, xerophyte and hydrophyte, right? So xerophyte live in the place like desert place where there is a lack of water. For hydrophyte would be the plants that can survive or that live or that they are adapted to live in water. So hydrophytes. And identify feature T shown in figure 5.1, and you need to explain why they adapt the leaf to float on the surface of the water. So when we look at the feature T provided, so it's like a airspace, right? So that's why I answer airspace. And for the explanation, so it's allowing gases to trap in there, reducing the density. So that's why it's float on the surface of the water and it gives the leaf buoyancy, right? So that's how the leaf float on the surface of the water if they have a lot of air spaces. And we need to explain other adaptation of this group of aquatic plants. So beside having a lot of air spaces, so we also learn about that they have thin cuticle, right? And they also have a lot of stomata, which can be found on the upper side of the leaf to allow gas exchange, since the lower part of the leaf is in touch with the water, which is very insufficient for them to have the gas exchange as well as for the transpiration, right? Even for the thin cuticle, it's also the part of the process for transpiration as well. Okay. And moving on to question number 6, which is about the environment of microplastic. They mentioned that microplastic are pieces of plastic with a diameter less than 0.5 centimeter. So I highlighted that. Okay, why do they give this information? So every sentence and every information matters while doing the questions in the exam because it's already giving you a hint. And figure 6.1 showed the mass of microplastic in the oceans between 2000 and 2040, and the data between 2000 and 2020 is an estimate, and data after 2020 is a prediction. So it's already mentioned there. But when we look at the graph, as the year increases, the mass of microplastic also increases. So this is the first information that we can get directly from the graph provided. And the question asked, suggest why the mass of microplastic between 2000 and 2020 is an estimate. So it's not accurate, right? It's an estimate. So why it is an estimate? So the information given there is it's less than 0.5 centimeters. So we can think of that. Microplastics are very small and they will be widely dispersed or like they will be widely spread in the ocean. So which make them difficult to measure accurately, or it's difficult for us to pick it up and count and like record and everything. It will be a very difficult work. And also, it is not easy to collect the exact data from all parts of the oceans, right? So ocean is a very vast area and we cannot, collect every single part of the ocean. And another thing that I think of it that the experiment might be carried out at different time or different location, or there can be other differences which might not be an accurate data or accurate result. So that's why this could lead to an estimate data for the mass of microplastics between 2000 and 2020. And using figure 6.1, you have to calculate the predicted percentage increased in the mass between 2000 and 2040. So as I look at the graph for 2000, it is around 0.2, and here, right? Here. And 2040 is around here, so which is about 1.7. So and I plug into the formula of like difference divided by original into 100. So the difference 1.7 minus 0.2 and divided by original, which is 0.2, and I got the percentage of 750%. And then you have to define the term producer. So the producer is an organism that can make its own food by converting the energy from the sun, or like converting the light energy to chemical energy, which is in the form of glucose or the carbohydrates by photosynthesis. And usually producers are the first organisms that can be formed in a food chain or a food web. And they show you a shearwater bird and its feed on fish in the ocean and suggest how microplastics can end up in consumers such as shearwater birds. So what I think is that the fish might also accidentally consume microplastic, right? Since they are very small and they might mistaken with their food and they might consume it. And so that microplastic will remain in the body of the fish and that fish might be eaten by the bird. So it is in the form of bioaccumulation and biomagnification, which passes along the food chain and it could ended up in the consumer or especially in the apex predator or top predator, right? Like humans like us. So we might also ended up consuming a lot of microplastic through the food chain, right? So that's how I come to this answer point. And we have to describe three ways that population of shearwater birds can be conserved. So it's a conservation program. So the first thing that came to my mind is to protect the nesting site or nesting area for those birds where they can, freely survive without any hunting or without any pollution. So which would be the protective nesting area. And also another thing we came up it's through the education where we can increase the awareness or where we can share the importance of the shearwater birds through the form of educating people or like through education. And another thing is captive breeding program where a zoo play a role in the captive breeding process where we can, conserve the population of shearwater birds. And lastly is to explain the risks to a population if its population size decrease. So imagine, right, when the population size decreases, it reduces the genetic variation, right? It reduces the number, so it reduces the variation. So when there is a less or when there is a reduction in the genetic variation, it would make the population less likely to be able to survive to the changing environment because there is a reduction in genetic variation. On the other hand, when there is a decrease in the population, so let's say the number of the population decreases, so it would lead to the less chance of mating, right? So it's how do I say? It's like there is a less individual to mate. So I answered that fewer individual available, so it could lead to inbreeding. So as a result of inbreeding, it can, weaker the variation in the offspring. Okay, so I think that's it for this paper and hope this help you in some form. And if you have better if you have better answer or if you have better approach to these questions, you can also, share back to me through the comment section or like, any form. Like I'm very welcome to your idea as well because these are just purely my sample answers to these questions and hope this help those students who are answering this major or in the later years as well. And thank you so much for watching my video and hope to see you all in next video as well. So have a great day and bye bye all. And again, thank you so much.